Welcome back, guys. Today we are going to talk about the plasma membrane and its permeability, or the ability of things to get through it. At the end of today's lesson, after the video, I'm expecting you guys to work on a reading guide that will be attached. We pretty much don't need to spend a ton of time talking about that because you all know how those work. I know that it may take you a little while to get through it, so I'm not going to be making this reading guide due, like actually due, until tomorrow, even though it's gonna say due today on there. That's just because I know you guys won't work on it otherwise, okay? I know you, I know how you do, okay? So let's talk about the plasma membrane. And if you notice, it's not a smooth little outside like we tend to draw them. It's kind of knobby and weird. This is why they look like actual boogers when we see them under a scanning electron microscope. So there is a whole lot going on here. So let's talk about it. So remember in Unit 1, we talked about phospholipids, that they had a phosphate group at one end. There's a glycerol and then two fatty acid tails. So it's not a triglyceride. It only has two little fatty acids right there. Okay, so A, what's that? To think for a second, that's our, hold on. So A is our phosphate group. We see we have the PO4. Okay, B is our glycerol right there. C, the whole C, is our hydrophilic head, because remember, one part of a phospholipid likes water and the other doesn't. So the tails are hydrophobic. So you might want to take a second and write this in your notes. You're going to need to know about phospholipids, okay? Okay. So the plasma membrane separates the internal cell environment from its external environment. It's mostly phospholipids. So we can see in this little picture, it's mostly those weird little brad looking things, but we know there's other stuff there. So phospholipids are amphipathic. Ooh, that's a fun word. So amphi or ambi, I don't think we've gotten to that prefix yet, but that means both. So like an amphibian needs both land and water, or an amphibious vehicle in the military can drive on the land, but it can also go into the water. So it's amphipathic. That means that one end is polar and one end is not polar, okay? So this forms a bilayer. We know that bi means two from our prefix list, okay? So that means there are two layers. It's not just one side. We have this double-sided business going on here. So selective permeability, the ability of a membrane to regulate substances that enter and exit. A cell doesn't want to let everything in all the time. It wants to control that. So the way I think of this, like a selective school only allows certain individuals in at a time. So they get to pick and choose. A cell membrane has ways of picking and choosing. Naturally, there's not an actual brain there, but it can, there's different chemical pathways it can use. Like if something's present, it opens up a chute, kind of like those chain reaction machines. So the hydrophilic heads are oriented toward aqueous environments. We see that AQU prefix there, that refers to water, okay? Hydrophobic tails face inwards and away from these environments. So a quick aside, actually soap behaves a bit like phospholipids do. They have a polar side and a non-polar side. And so that non-polar side is going to bury itself in oil or whatever, and then the hydrophilic part is what lets you wash that away. That's why if you get grease on your hands and you put it under just water, you can't get the grease off. But if you use soap, the hydrophobic parts are going to surround the oil molecules because they can, like non-polar dissolves in non-polar. And then the outsides are polar, so they can face the water. So yeah, that's also why you might see like little beads of oil in the dishwater anyway. Anyway, I'm sorry, saponification. Anyway. Okay, so the fluid mosaic model, this is what we use to describe our cell membrane. It's not like a rigid plastic bag. These little bits and pieces move around. These little brad-looking phospholipids, they can shift and move around. The channel proteins can move around a fair bit. So they're held together by weak interactions and they can therefore move and shift, and this can be a good thing. So temperature affects fluidity. They are lipids after all. If you put butter in the freezer, it gets super hard. If you leave it at room temperature, it's soft. So this just, it makes intuitive sense that temperature can affect fluidity. So the unsaturated hydrocarbon tails help maintain fluidity at low temperatures. So remember that you have to get unsaturated fats colder than saturated fats to get them solid. Follow my thinking for a second, okay? Think again of butter. Butter, even though it gets soft at room temperature, it still holds its shape. If I put a stick of butter on the counter, it's not going to melt unless it's August and your air conditioning's out, okay? But normal circumstances, that butter's going to keep its shape. It's going to stay a solid. Olive oil on the counter stays liquid. So it's more fluid at lower temperatures. Like I have to actually heat that butter to melt it, whereas the olive oil is already liquid. You have to literally put the olive oil in the fridge to make it solidify, which happens, by the way. Yeah, so those unsaturated hydrocarbons, that's what keeps it fluid, just like unsaturated fats in food. Okay, and also, sorry about that bar. I don't know if y'all can see it, but it's bothering me. So that's why you see the little bent tails on there. If they were perfectly straight, it would be easier for them to lock up, but since they're bent, they get some more movement. Right there. Okay, so cholesterol. Cholesterol isn't all bad. Cholesterol is actually vital to your body's functioning. Your body makes cholesterol on its own, and part of its usage is it's another thing that helps maintain the fluidity at both high and low temperatures for your cell membrane. So at high temps, it helps reduce movement, like hold it together, and at low temps, it reduces the tight packing. So just, it helps keep that space so it's not locking up. And so what the word mosaic means that there are lots of pieces interlocked together. There's lots of different macromolecules involved. A mosaic in art is a picture made by lots of interlocking pieces that are different colors. So the mosaic model in a cell membrane, there's lots of different pieces involved. That's what they're getting at. So it's a fluid, meaning it moves around, and it's a mosaic, meaning that it has different parts put together. Okay, so membrane proteins, there's two major categories: integral proteins, ones that are embedded into the lipid bilayer, like transmembrane proteins, and then peripheral proteins that are not embedded into the bilayer and they're loosely bonded to the surface. So in this picture, we can see the ones that are like stuck all the way through. Those are going to be integral. So if you say that Mr. Dickey is integral to this school, he is a central part that keeps the school together, okay? Or a foundation's integral to a house. So it's in there, it's embedded in there, it's an important feature. So these integral proteins are built into the membrane. Like this term transmembrane, trans can mean like through, like a, like translocation, you're teleporting through to somewhere else. So it's going from one end to the other of this membrane. So these proteins are also amphipathic. If you remember that word, that means that it has both a polar and a nonpolar side, and that's important because it keeps it anchored in the nonpolar part of the cell membrane. And then peripheral proteins, those are those little branch-like ones on the outside of the cell. Okay, so there's also some carbohydrates, and this is going to be difficult and a little confusing because they're both carbohydrates and lipids at the same time. And before you throw your pencil at the screen, okay, remember, like a fatty acid, it can have characteristics of both and regions that do both, okay? So these molecules have both things attached together. So glycolipid has a carbohydrate attached to a lipid, and a glycoprotein has carbohydrates bonded to proteins. So it's pretty self-explanatory. So we can intuit from this that glyco refers to sugar. We remember glycogen from the macromolecule unit. That is carbohydrate storage in animals. So it's something in your liver that stores sugar. So it has that glyco word. And like glucose, or your glycemic index, if you're diabetic, has that same prefix in it. So it refers to sugar. So glycoproteins are the most common. Just a quick aside for that, when we get to cell signaling, these are going to be really important later because this is how one cell is able to recognize another, are these markers on the outside, and this is how antigens often get created. So I'm going to skip down to plant cells because it's ever so slightly different because they are a lot more together, if that makes sense. So instead of like freestanding houses like we think of animal cells, plant cells are like apartment buildings. There's a whole lot of them stacked right on top of the other. So plants also have a cell wall over their membrane. So it's extracellular and not found in animal cells. Extra means outside, like extraterrestrial is outside the earth. So extracellular is outside of the cell. And animal cells don't have cell walls, not typically. So these cell walls provide shape and structure, they provide protection, and they help regulate water intake. I'm gonna hold up, oh no, but I don't know if you can see it. There's a plant cell and an animal cell that my sister drew for me up there on my board. So plant cells tend to be a lot blockier. Like you see in this slide image, they're not weird booger shapes. They're kind of look like jewels a little bit. So these cell walls, they're important. This is also why wood is hard and why vegetables are crunchy, is because they've got these cellulose cell walls. So it's thicker than a plasma membrane, and it has something called plasmodesmata. Ooh, that's a big word. Okay, so those are holes in the cell wall so that other cells can be connected. This can be super important in plants. Just think about it, right? A plant has to get water from its roots all the way up to its leaves. So if there was no way to draw water from cell to cell, there would be no way to get that nutrition from one end of the plant to the other. They don't have a heart. So they have to do that by being able to transfer those things from cell to cell to cell. Kind of weird, but cool at the same time. Okay, guys, so that's it for today. I want you to get started on your reading guide. I know it says due today. I'm not going to be mad if you have to finish it tomorrow. That's kind of what we have planned. Also, tomorrow is your prefix test, so be ready for that 11 through 20, okay? And I will see you guys in the next video. Take care.